So in this video, I want to talk about lactose tolerance and intolerance. Okay, so lactose intolerance is the state where an individual cannot digest lactose. So individuals that we call lactose intolerance, so typically we're talking about adults that cannot digest lactose. Now, most of these adults could digest lactose as children. Okay, so here's a little bit about the biochemistry. So lactose is the sugar on the top, and it's digested by this protein, this enzyme called lactase, to produce these two sugars that then go on to be digested by further processes. So we talked about whether this is genetic or environmental, and I said that scientists have worked out that differences between lactose intolerant and lactose tolerant individuals mostly have to do with genetics. And then we talked about how much of the genome do we think is responsible for lactose intolerance. And then I gave it away. I told you that that other scientists have found that it seems like it's a single nucleotide that mostly distinguishes lactose tolerant and lactose intolerant individuals. So then we talked about how can this be the case? How can a single nucleotide influence a whole trait like lactose tolerance? Okay, so let's work this out. We said that lactase, this enzyme that digests lactose, is a protein, so that must mean that there's some lactase gene. And so if this is our lactase gene, let's figure out what's going on here. So in childhood, we get production of the lactase protein because we get digestion of lactose. And then in lactose tolerant individuals, that means we're also getting lactase in adulthood. So that means that our lactase gene is expressing mRNA and then making protein. So in both childhood and adulthood, we get DNA to RNA to protein, DNA to RNA to protein. Okay, so then what's going on in lactose intolerant individuals? Well, we call them lactose intolerant because they can't digest in adults, but we said that when they're children, they are producing lactase, which must mean that they're producing DNA to RNA to protein from their lactase gene or lactase, both their copies of the lactase gene in childhood. But in adulthood, what's going on? Well, we're not seeing production of lactase. Okay, so lactose tolerant individuals are doing the same thing in childhood and adulthood. Lactose intolerant individuals have this difference between childhood and adulthood. So then we asked, how could it be the case that a single nucleotide was driving this? And a lot of people came up with the same idea, that a mutation in the gene somewhere was leading to no functional lactase protein being produced. But then I challenged you and said, well, but isn't that surprising given that these same individuals can produce lactase in childhood? And so if it were the case that we had some mutation that was destroying the gene or disrupting the gene, we'd also expect the same effect in childhood, but we're not seeing that. So what's going on? So these scientists actually identified what the specific nucleotide mutation is, and interestingly, they did not find a mutation in the gene. They found a mutation about 14,000 nucleotides away, and it's this site right here. And what they found is that some individuals have a C and some individuals have a T. So let's figure out how this matters. So let's start with the individuals that have a C. So during childhood, these individuals express the gene. As we said, they make mRNAs, and then off to the right here, they make proteins. But I've left them off the slide. But what happens in adulthood is that individuals with the C, they have an OCT gene, this protein called OCT1. I shouldn't have said gene, sorry. They have this protein called OCT1, which comes and binds to this site, and it actually blocks production of mRNA from the lactase gene. So we get no transcription, so we get no translation. Okay, so just think to yourself, are these individuals lactose tolerant or lactose intolerant? Okay, well, they don't have lactase as adults, so that means they cannot digest lactose, so these are individuals that are lactose intolerant. Okay, so let's think about the individuals that have a T. So childhood, it's the same deal, but then in adulthood, though they have functional functional OCT1 proteins. Those proteins cannot bind to this binding site, and therefore they cannot block production or expression of lactase from this lactase gene. And so these individuals in adulthood have the same expression that they had in childhood. So these individuals, because they're expressing lactase in adulthood, are lactose tolerant. We call them lactose tolerant. They can tolerate lactose, they can digest lactose in adulthood just like in childhood. So I wanted to talk about OCT1 expression in adults. So this was funny when I searched this on Google. It said it's OCT1, it's October 1st, and adults can now legally buy weed. So great. Okay, so then I eventually found this though. And so let me just show what you're looking at here. So this is a database where scientists have figured out expression of each gene, in this case OCT1, in all these different tissues and in all these different stages. So this is in mice, so these different stages. So here we have adults, and then these are different embryonic stages. And so what you see for the most part, OCT1 is not expressed early on, and it is expressed late. And particularly we want to look at the gut because we're talking about digestion of lactose in the gut. And so what you see is that OCT1 is not expressed through all of development, and then it is expressed in the gut in adults. So this makes sense. So we said that OCT1 was on in adults and off in children or in embryos. So this means that OCT1 itself is regulated. Okay, so let's work this all the way through. So in childhood, OCT1 is off; it's not expressed, and so that means that nothing binds here to block lactase expression, and so lactase is expressed. So in lactose intolerant individuals, to start out with, so OCT1 is expressed during adulthood, and it comes down and it binds, and it blocks expression of the lactase gene, and so we don't get any expression of lactase. So in lactose intolerant individuals, we have no lactase in adulthood. Okay, so what about lactose tolerant individuals? So in these, so during childhood, OCT1 is not expressed, so there's no binding to block lactase expression, and so we get expression of lactase in childhood. In adulthood, we get expression of OCT1, however, it cannot bind to this negative regulatory site, this site that negatively regulates lactase. So there's nothing to bind here and block lactase production, and so lactase is produced, and we get production of lactase proteins. So this is the general concept of regulatory networks. Here we've been talking about how OCT1 regulates lactase, although we point out that OCT1 itself is regulated. And so what we're looking at here is, so each one of these things would be a gene that's producing a protein. And so this would be like, if this were OCT1, it would be regulating lactase. And this red bar would mean that it's negatively regulating lactase, just as we've expressed, just as we've discussed. But OCT1, we said, is itself regulated, so that means something before it is going to be regulating it. So in this case, we've got some other gene producing another protein that's negatively regulating, and that's negatively regulating. And so in general, we have green meaning positively regulating a gene, and red meaning negatively regulating a gene. And so these things can get quite complicated. So here we have a whole regulatory network for bacteria, and what we see is within this network, we have regions that are responsible for genes or that involve genes that are responsible for responding to cold shocks or formation of communities, biofilms, or response to different drugs. And so these networks can get quite complicated. Quite interesting stuff. Okay, but let's return to lactose and lactase. So this is a map of lactase persistence. Lactase persistence. Okay, let's stop on that for a minute. What do we mean by lactase persistence? So to persist means to continue. So actually, lactase persistence means having lactase expression in adulthood. So that means lactase persistent individuals are individuals that are lactose tolerant because they can digest lactose in adulthood. And lactase impersistent individuals are those that do not express lactase in adulthood, and this cannot express lactose. Sorry, cannot digest lactose. Okay, and so what we're looking at here is a map of human populations, and we're looking at the frequency, the fraction of people that are tolerant to lactose. And so what we see is through much of the world, through all of Asia, through Australia, and through most of Africa, a large majority of the people are not tolerant to lactose. So they're lactose intolerant. They're down at this end of the spectrum. And it's only in Europe and then also here in West Africa that we have peoples who are typically lactose tolerant. They're at the high end of the spectrum. Okay, so what we're going to do now is we're going to look at the NCBI, the National Center for Biotechnology Information, the SNP browser. So, okay, what's a SNP browser? So a browser, you know what that is. A SNP is a single nucleotide polymorphism, just the kind of difference we've been talking about, where we have one nucleotide that's different between different copies of a gene. So let's go ahead and have a look at this database. And so here, so this is basically the web page for this particular change that we've been talking about. So you can see here it's a CT difference. Probably here somewhere it says, it doesn't say that. So it's a CT difference. So we can go through some things here. There's a whole lot of information, but I'll just highlight a few things. So organism, so it's in humans. It's in the genome. It's a DNA difference. So then we have the variation class. So it's a single nucleotide variation. This relates to when we were talking about, are the differences here, is it the whole genome? Is it a whole gene? And we decided, no, it's a single nucleotide difference. And it's a difference between some individuals have C, some individuals have T. So this tells us about sort of the position. So this is telling it's on chromosome 2. We'll talk more about chromosomes later, but of the 23 huge molecules that we each inherit from each of our parents, this is the second longest, what that means. And it means within this chromosome, it's actually a position, if we counted from one end of the chromosome, it would be a position 135,859,184. So this tells us what gene it is. So probably if we clicked in there, let's go ahead and do it. Uh-oh, this is not going to make me look good, I don't think. Yeah, so this is summary. Protein is a gene, one of the. So it's a little bit more complicated than I thought it might have been, but they talk about how it's neighboring the lactase gene, this is talking about the involvement of this in expression of the nearby lactase gene. Okay, and we can continue down. And so the part I really want to highlight here is this part. Okay, so what are we looking at here? So each one of these lines is giving us a readout of how frequently individuals have either blue or red, how either the C allele or the T allele. We said there are two different possibilities. You can either have C or T at this site. And what I want to focus in actually on here is this group. So this is East Asian, Europeans, Africans, Native Americans, and I'm actually not sure what this fifth population is. So let's start out with this. In East Asians, we see that 100% of individuals have the blue. And we can go back up here and remind us that the blue is the C. So the C is the lactose intolerant, the individuals who turn off lactase in adulthood. And then in Africa, we see a similar thing. But interestingly, in Europeans, as we were discussing, about half of the alleles that we find in Europeans are the T, which we said was lactase tolerance. So I just want to point out here that here we're looking at a database where people have sequenced the DNA of large numbers of individuals, of 1,000, of 1,000, of 1,300, of 700. And what they've found is that there's this difference in the DNA, which exactly reflects what we see when we look across the population. Of course, it would be great if we had 1,000 individuals from these populations, 1,000 from these, then we would really see the difference. But in general, we see very little phenotypic evidence for lactose tolerance in Asia, and we see very little of the allele. We see just a little bit of lactose tolerance in Africa, and we see just a little bit of the allele. But in Europe, we see a lot of lactose tolerance, and we see a lot of the allele. Okay, so that was a lot. I just want to go through one more thing, which is this concept of dominance. So what do we mean by dominance? So we've sort of seen it so many times that we've forgotten almost how we define dominance. So what we mean by dominance is when heterozygotes, people who have one copy of each, have the same phenotype as one of the two homozygotes. So what we mean here, so in this case, people with two C alleles are intolerant. So individuals who have two of the intolerant alleles are intolerant. People with two T alleles are tolerant. And so then when we're talking about dominance, what we're talking about specifically is what do we see in terms of tolerance in individuals that have one copy of each of the two alleles? So let's see if we can sort of reason this out and think this out. So now we're talking about individuals that have one of the C's and one of the T's. Okay, so in childhood, there's no OCT1 allele, so actually we expect both alleles to be expressing proteins, to be expressing mRNAs and expressing proteins. Okay, so but in adulthood, now we do have OCT1 protein, so that OCT1 protein is going to bind to this allele and block expression from this lactase gene. However, as we've discussed, this OCT1 cannot bind to this site, and so this copy of the lactase gene is going to be on, and we're going to be expressing lactase. So basically, we're going to get roughly half as much lactase as we otherwise would. And so then the question basically is individuals that have half as much lactase, heterozygotes who have half as much lactase, what is their phenotype? Can they digest lactose or can they not? And so what we end up seeing is that having half as much lactase, that's enough to get the job done, to digest lactose. And so what that means is that these individuals have one C and one T. They are lactose tolerant. They can digest lactose, almost just as though they were TT homozygotes. So this is, just to point this out, this is the definition of dominance, that the heterozygotes look pretty much just like one of the two homozygotes, that they don't look kind of halfway in between. They are lactose tolerant, and that's what we mean by dominance. And I just want to point out that this is frequently the case. It is frequently the case that if you have one allele that is expressing the protein and the other allele that is not expressing the protein, which means that you probably have about half as much of the protein, that's often about just enough. It's enough to get the job done. So this is commonly the case. Half as much protein is enough, and so you end up with the allele that's expressing the protein being dominant over the allele that is not. So we'll be talking about this more, but I just wanted to get us started that dominance is not some mysterious abstract concept. It arises from this fact that often when you have half as much of a protein, that's enough to get the job done. Okay, so to finish up, key concepts that we talked about are this concept of gene regulation. As we'll learn, this can be negative, as we discussed here, or positive. These binding sites of the regulator, where they bind near in this case near the lactase gene, a mutation in the binding site can change the gene expression because it changes binding of the regulator. And finally, we can often predict dominance or recessivity of mutations.